Hola, I'm Miki Ebisuya, a group leader at EMBL Barcelona. I'm going to introduce one equipment in the lab, which is a microscope that was transported all the way from Japan. 2 years ago, my lab moved from Kobe, Japan to Barcelona, and because this microscope is custom-made, we've decided to ship it to Barcelona. So the microscope safely arrived in Barcelona, and this box is actually an incubator to keep the temperature and CO2 concentration constant. And inside the incubator, there's a microscope, and we can culture up to 8 samples that contain living cells to take movies. And here's one example movie that was taken by this microscope. In the movie, there are several hundreds of cells, and the color indicates protein amounts of one gene. As you see, the protein amounts goes up, down, 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 up, down. So it's oscillating or fluctuating. Here's a quantification. The x-axis is the time, y-axis is the protein amount, and it's oscillating. This oscillation or rhythm is important for the formation of repetitive structures in our body, such as the vertebrae. And interestingly, recently we have found that this oscillation, the speed of this oscillation, differs between species. In the movie, on the left are mouse cells, and on the right are human cells. And both samples show nice oscillations. However, in mouse cells, the protein amount goes up, down, up, down, up, down, up, down, up, down, up, down. But in human cells, the protein amount goes up, down, up, down, up, down, up, down, up, down. So the human oscillation is slower. Well, actually, one oscillation cycle takes 2 to 3 hours in mouse, but it takes 5 to 6 hours in human cells. And now we are studying why the speed or tempo is different between the species. Thank you.